inflammation, basically, it is the response of our body towards any injury, towards any harm that the body face in terms of any trauma, in terms of any accident, or in terms of any infection. So the reaction of our body is known as inflammation. Here you can see that a big toe of a person is hurt. So what happens? The inflammatory reaction starts occurring, and there are different changes which are very visible. Like there is a swelling, there is a change in color of the skin, and many others. So the simplest and the basic definition of inflammation is the response of human body against any injury. This is the easiest definition that you can remember, and then you can say whenever someone can ask. So there are two types of inflammation, that is acute inflammation and the chronic inflammation. Causes of inflammation are very important, that how inflammation occurs. There are a lot of causes, among them like infective causes, like any infection that occurs inside the body. It could be bacterial, viral, fungal, parasitic, and any toxins, microbial toxins that is caused. Then immunological agents. Physical agent is usually the physical injuries, traumas, accidents, burns, and any um, okay. Immunological agents includes hypersensitivity reactions. Usually what happens whenever our body in which the normally we have a protective immune system. Whenever our immune system damages our own body in case of autoimmune diseases, so what happens is inflammation starts occurring inside the body, and the body starts reacting against it. Then chemical agents, sometimes persons come across with different injuries, chemical burns, accidents. So what happens that there are different chemicals that can cause inflammation. Similarly, there are some foreign bodies or the inert material, like cholesterol crystals in case of atherosclerosis, and smoke particles in smokers. So all these are the causative agents that can cause inflammation. The signs of inflammations are very important. Whenever a person come across with inflammation, he or she has got local change in color, that is called the hyperemia or rubor. Then again, the temperature of that area is elevated. That causes fever, that is called calor. Then tumor basically is a swelling, the area swells up. Then pain. Then sometimes if the inflammation is very extensive, or in case of chronic inflammation, the particular area of the body part loses its function. So that's called functional impairment or functiolysia. So these signs of inflammations are very important. One should know that what happens when inflammation occurs. Well, the main features of acute inflammation, basically what happens that there is increase in permeability. The arteries or the veins, they have got increased permeability, so the cells, they escape from the diameter of the arteries and the venules, and they enters into the extracellular matrix or the extracellular spaces. So what happens, that area becomes swells up. You can see that in normal, what happens that arterioles have and the venules both have a specific diameter, and the extracellular matrix has got a specific concentration. But in case of inflamed area, what happens that the neutrophils, um, leukocytes, they emerge out of the vessels and they enters into the extravascular space, and it initiates edema. So ini uh, so it all causes a swelling. So this is the way how inflammation starts. Recognition of invaders. So what happens whenever our body come across with any foreign uh, um with any foreign particles or the microbes, how our body recognizes those aliens, those invaders. So we have got receptors. Our body is blessed to have different receptors. Some receptors are present in the plasma membrane, some that are called extracellular receptors, some are present in the intracellular cytosol, then endosome. So what happens, these receptors, they recognizes the any foreign invader, and they get attached to that microbe or any foreign particle. So these are the cellular receptors. Our body has got sensors of cell damage. So in usually what happens that our body undergoes wear and tear mechanisms. So what happens that whenever our cell damages, whenever our DNA damages, there are increase of certain substances in our body. These substances are ATPs, uric acid, and low concentration of potassium ions. So what happens, all these are the sensors. When the body undergoes such changes, it means that there is something going wrong with us, with wrong with our body. So the inflammatory reactions are going to start it. We have some inflammasomes that are they are the receptors, systolic receptors. Basically what they do, systolic receptors are intracellular receptors. So what they do, they activate some inflammasomes, especially interleukin ones. So interleukin ones, they trigger the leukocytes, they stimulate the leukocytes to reach to the inflammatory process, to the inflammatory places, or the place where the foreign particles have been invaded, and they alert the leukocytes to go and kill them. Other receptors are some leukocytes, they have receptors for FC. FC is the constant part of the antibodies. You know, we have a very well developed mechanism of identifying microbes. Whenever our body come in cross with any microbes, the antibodies are formed by the B lymphocytes, and these antibodies, they attached to the entering microbe. So what happens, the FC part, antibodies has got two parts, that the constant part and the variable part. FC is the one who is the constant part. So FC portions of the antibodies, they attaches to the leukocytes or the WBCs. So antibodies are already encoded with foreign particles, and the FC part of the antibodies, they attaches to the leukocytes and make them alert about the foreign particle. Moreover, there are some circulating proteins like mannose binding lectin receptors, like complement system. They are circulating in our body, and whenever they found any foreign body, so they attaches themselves and make them available for the immune system for further killing. So this step, that is the recognition of invaders through different receptors, is very, very important. Acute inflammation has got two events, or we have been divided into two parts, that's the vascular events and the cellular events. In vascular events, what happens that there are some hemodynamic changes. Initially, what happens whenever body come across with any injury, there is initial vasoconstriction. Then what happens after few, you can say seconds or minutes, this vasoconstrictions uh, converted into vasodilation, right? So what happens? The diameter of the arterioles and the venules becomes thicker, and there there is progressive vasodilation. Like once the vasodilation has been started, it keeps on going due to the increased hydrostatic pressure. Then what happens that due to the vasodilation, it will eventually leads to the increase in permeability, and all the proteins and all the WBCs, they leave from the vessels and enters into the extracellular spaces. So what we have inside the vessels are RBCs. So the RBCs, they get attached to each other and formed a stasis like appearance. So due to this, there is increase in viscosity of blood because all other cells and all other particles have been migrated to outsides. Only RBCs are left behind. So these RBCs, they causes stasis or increase in viscosity of blood. Increased vascular permeability, basically it is caused by contraction of the endothelial cell. You know that the vessels are composed of different endothelial cells. So what happens that the endothelial cells, they start contracting. When they start contracting, what happens? There is appearance of gap between the two endothelial cell, and this gap will allow the um, exudation of fluid from inside of the vessel to the outside of the vessel. So there are two type of responses. First is immediate transient response. Actually, this is 15 to 30 minutes. It is miswritten over here. This is not second. It is 15 to 30 minutes. So immediate transient response occurs after 15 to 30 minutes, and delayed prolonged leakage occurs after 2 to 12 hours. So that's a pretty long time period. And basically, the contractions of endothelial cells is due to some chemical mediators like histamine, bradykinin, and leukotrienes. The delayed response may be caused by contraction of endothelial cells, or it could be due to the mild damage. So, late appearing sunburn is a good example. Endothelial injury mediated by leukocytes, direct damage to the endothelial cells is oftenly found in many injuries. For example, in burns, what happens in burns? The blood vessels are directly damaged, and neutrophils usually adhere to that um, damaged vessels. Actually, in case of normal healthy endothelial lining, the leukocytes will not uh, get attached to the endothelial linings in case of healthy endothelium. But in case of endothelial injury, what happens? The leukocytes, they forming attachments um, along with the injured endothelial cells, and they further start, or they further enhancing the injury of the endothelial cells. So what happens due to this? There is increased exit of fluids and proteins called the transcytosis through the endothelial cells, and this basically involves the intracellular channels uh, that are activated by certain factors like vascular endothelial growth factors. Response of lymphatic system. Our lymphatic system also plays a very important part during the inflammatory process. Usually what happens that most of the time excess fluid in extracellular spaces are drained by the lymphatic vessels. But in case of inflammation, what happens? The lymphatic vessels and the lymph nodes, they become inflamed itself, they become damaged. So the two conditions arises, that is the lymphangitis and the lymphadenitis. So this is the mechanism. We will discuss that how increased vascular permeability occurs. Usually there is a balance between the hydrostatic pressure and the colloid osmotic pressure. But in case of vascular permeability, when it is increases, the balance is totally out of balance. And what happens? There's increased hydrostatic pressure and the decreased colloid osmotic pressure. I'm sure that you all know about the colloid osmotic pressure. Colloid osmotic pressure is a pressure which is exerted by the proteins that are present. So what happens when there is increased
हेलो डॉक्टर फौजिया, आपकी आवाज नहीं आ रही है।
अब आ रही है?
जी अब आ रही है।
अच्छा, ये माइक का मसला है गैस। अच्छा, what happens in case of increased vascular permeability? There is imbalance between the hydrostatic pressure and the colloid osmotic pressure. Colloid osmotic pressure is the pressure which is exerted by the proteins that are present inside the fluid. So what happens when there is increased endothelial spaces and the vasodilation occurs? The proteins and the fluid, they leak outside the vessel, and the colloid osmotic pressure decreases, whereas there is increase in hydrostatic pressure. So whenever there is high protein content outside the vessels or in extracellular spaces called exudate, and when there is low protein content or increased hydrostatic pressure, so that calls the transudate. So in some diseases where protein is lesser in amount, like in liver disease, like in kidney disease, like in protein malnutrition, for example, in kwashiorkor. So there is a condition called transudate occurs, or in case of congestive heart failure. So this is, we have already discussed what is the exudate, transudate, and the edema. In case of cellular, even what we have exudation of like leukocytes and the phagocyte. Exudation of leukocyte means that leukocytes, they exit out of the vessels. It is consisting of four stages. That the margination means they margin themselves along the endothelial lining. They move in a rolling pattern, then they adhere to the endothelium, and finally they leave the endothelium through the interendothelial spaces. These are the events. So first there are two important things we should understand. These are the integrins and the selectins. Integrins are found on leukocytes, and they have lesions as well. Lesions are very helpful in enhancing the function of the integrins. So in case of selectin, what happens? There are different types of selectins, like P selectins, which expresses on platelets, then E selectins, which are expressing on endothelial, and there is L selectins that are used for leukocytes. So what happens that integrins that are present on leukocytes, first of all these leukocytes, they arrange themselves along the margins of the endothelial vessels, endothelial lining. Then what happens that these integrins, they attach to their respective lesions on the endothelial lining. In this way, you can see this is E-selectin, and the lesion of E-selectin, that is sialyl Lewis X modified glycoproteins, are found on the leukocyte. So what happens when they arrange themselves along the endothelial linings? So their legions of selectin on leukocytes, they get attaches on their respective selectin, and the integrins also, they attaches to their respective lesions. So in this way they walk along, or you can say they roll along the endothelial lining. This is very important to understand that integrins and selectins are the most important, you can say, receptors for the exudation of leukocytes, right? So after moving along or rolling along the endothelial linings, what happens? They move through the gaps, inter endothelial gaps, and they escape out of the vessels. This is the vessels, right? This is the rolling mechanism. Then their adhesions, when the selectins and integrins, they attaches firmly with their lesions, they get stable, and then they migrate through the endothelium because of the interendothelial spaces. And these PCAM or CD3 ones, they are very helpful in letting them out from inside of the uh, lumen of the vessel. When they came out, so they move towards the site of injury, and that's called the chemotaxis. There are certain chemokines, and there are certain proteins which act as, you can say, they attract these leukocytes towards the site of injury by the process of chemotaxis, in which C5a and 3CB is very important. So in chemotaxis, basically what happens, they are sometimes the complement system, like I have said, C5a, cytokines, that is IL-8 or interleukin, and certain toxins that are releases from the microbes are very important. As well as there are certain arachidonic acid metabolites that are very helpful in chemotaxis. The question is that we have different leukocytes. Neutrophils are the one who are responsible for acute inflammation only, and the macrophages are found in chronic inflammation. But the question is why the neutrophils are appears in first in acute inflammation, why they are not present in the chronic inflammation. So the answer is basically that the neutrophils are greater in amount, and they have more activity towards, or you can say, they are more rapidly responding to the chemokines, and they have good um, addition ability to the addition molecules. So that's why they are more frequently found in acute inflammation, and the most important thing, that they are short lived. They can't live for a longer period of time. That's why once the acute inflammation changes into the chronic inflammation, they completely die. Phagocytosis is basically is a process of foreign engulfment of the foreign body. Like in this process, the bacteria are recognized by the body or the immune system of the human body, and they are engulfed, and finally they are killed. So the stages of phagocytosis comprised of, first, the recognition and attachment of the particle, then engulfment, and then killing. Let's discuss in detail Okay, I have just told you that the phagocytic cells are neutrophils that are free early onset, and the monocytes and macrophages. Basically, monocytes are free living, and when they are fixed, they are converted into the macrophages. So, what happens first of all? This is the microbe. Kindly pay attention and understand this mechanism. It's very important. This is phagocytosis. What happens that the microbe is recognized by phagocytic receptors. What happens that the phagocytes like macrophages, like monocytes and the neutrophils, they have receptors. They recognize the or um, um, they capture any microbe through these receptors. It means that the microbes have to bind to the receptor. If the receptors are not working properly, so microbes cannot be killed. So these are the receptors. So this is microbe. Microbe has already got receptors, and these receptors they come in contact, and the microbe attaches. Finally, what happens when the neutrophils, monocytes, and um, macrophages they recognize the microbes through the receptors? They engulf it. In this way, a phagosome developed and they pulled the microbe inside this phagosome. After engulfment, uh, this complete vacuole is formed around this ingested microbes. Then what happens that this vacuoles has got different killing enzymes like the lysosomes and different phagolysosomes. So these enzymes start digesting this microbe. So finally, what happens that they start killing. This process is the killing and degradation. In what happens that there is a complete system of killing mechanism. Like there are certain enzymes that are present in these lysosomes. So what they do, they produce reactive oxygen or reactive nitrogen species. So these reactive or nitrogen species, they are killing. They have the ability to kill microbes. So in this way, these microbes are killed. Like there's an enzyme called NADPH oxidase that convert NADPH to NADP. So this when this happens, so the oxygen converted into the free radicals, and the free radicals have the ability of they are the ability of microbicidal activity. So in this way, this mechanism perform their function of killing microbes. Okay, phagocytism first recognition and attachment. As I have told you that there is different receptors on macrophages, like presence of opsonins. Presence of opsonins basically are those substances that attaches to the microbes and make them palatable, or you can say that make them eatable for our immune system. So there are different opsonins like IgG and C3b. So these are the different options. Intracellular destruction that occurs inside the phagosomes are, I have told you that reactive oxygen species, reactive nitrogen species, lysosomal enzymes like lysosome, collagenase, gelatinase, acid hydrolases, myeloperoxidase, as well as some neutrophil extracellular traps are also present. Leukocyte mediated injuries, which usually occurs in autoimmune disease and allergic reaction. So there is a well established mechanism present, which helps in killing. Well, so far we have been talked about the procedures of the acute inflammation, but what happens if the acute inflammation is going to be terminated? Either it is the removal of the offending agent, like the offending agent has been gone, there is no offending agent right now, so the acute inflammation will be turned off, or the neutrophils will die, or there is some cholinergic discharge that occurs inside the body that stops acute inflammation, and sometimes there is a self-triggering mechanism. Sometimes our bodies start to stop inflammation. That is the self-triggering mechanisms of anti-inflammation. Well, mediators are that are plays very important role in inflammations, that starts inflammations and they regulates inflammation are cellular, plasma, and necrotic cells. So these are the principal mediators. That is very important that you should know the name of some very important. Whenever I say something very important, that it is important from examination point of view. So this table is very important, in which the histamine, prostaglandins, leukotrienes, they are very you should know their mechanism of actions or mass their sources as well. So this table is very important. Arachidonic acid metabolites and their role in inflammation is also very important. We cannot proceed without discussing this. So basically, arachidonic acids are formed from the cell membrane phospholipids. I'm sure that you have an idea about the structure of the cell membrane. You have studied in physiology. So just recall a bit that there are some phospholipids that are found in the cell membrane, and they converted into the arachidonic acid through phospholipases, right? Then further, acid has to undergo two mechanisms, that is the cyclooxygenase and the 5-lipoxygenase. Cyclooxygenase is moving towards the formation of prostaglandins. We know that prostaglandin is very important. It is not new for us. So each prostaglandin has got different role, like prostaglandin G2, H2, I2 that causes vasodilation, inhibit platelet aggregation, thromboxane A2 that causes vasoconstriction, then the D2 and E2. Uh, we will have a question and answers session at the end of this lecture. Just concentrate on it. Another mechanism is 5-lipoxygenase. It is associated with the chemotaxis, and it also has different types like A4, C4, D4, E4 that causes bronchospasm, increased vascular permeability. And then we have 12 lipooxygenase pathway, which produces lipoxygens A4 and B4, which eventually inhibit inflammation. So this is the production of arachidonic acid metabolites, and they played a very important role in inflammation. Principal mediators of inflammations are again very important, like histamine and prostaglandin are associated with the vasodilation. Histamine again and serotonin, C3AS and C3B, leukotrienes, they are associated with increased vascular permeability. Chemotaxis are linked with C3A, C5A, leukotrienes, B4, fever, pain, tissue damage. So these are very important that you should know the principle, because in the book there are a lot of mediators are given, but don't focus on that on too much detail. Like what I have given over here, I have just chunk out the major and the very important mediators that are associated with the inflammatory processes and they and their reaction during inflammation. So you should know that, because it is very, again, I'm saying that they are very important for the examination point of view. Like morphological pattern of acute inflammation. So far we have studied the chemical processes that occurs during the acute inflammation, but now what are the morphological pattern? It is divided into three, like serous, the body cavities, that is the pleura, peritoneum, pericardium, they are lined with the serous fluid. Then fibrinous, in which there is a large quantity of fibrinogen. And then the purulent, or the pus, or you can say there is a formation of abscess, in which there is a increased quantity of neutrophils, necrotic fluid, and edema fluid. So what happens when the acute inflammation subsides? In acute inflammation, we have studied that there are vascular changes, there are neutrophil equipment, and certain mediators, right? What happens? Either it is progresses, if it is not treated properly, it is, if the triggering factors is still there, so it will be progress into chronic inflammation. Or if the stimulating factor is benign, it is not very malignant, so it could be healed. Or if the trauma or the causative agent is in very small amount, it is not that much devastating, so it could be healed. It could be healed simply, or it could be healed by forming abscess, or it will resolute, and the function of that part of the body will be resumed. So these are the different fate of acute inflammation. Chronic inflammation, basically, I have told you that it is consisting of macrophages, main important cells, lymphocytes, and the plasma cell. There is a tissue destruction or healing. Usually what happens that in case of chronic inflammation, there's a greater amount of destruction in tissue. Causes could be persistent infections, prolonged exposure, or the hypersensitivity diseases. Hypersensitivity diseases are those that are linked with our immune system. When our immune system is not working well, it will start causing uh, harm to our own body. So it will start leading to chronic inflammation. In When we talk about the macrophages basically they work through two pathways. That is the classical activated macrophages, that is directly activated by the microbes. So what happens that macrophages has certain mannose uh, you can say that mannose particles. That mannose particles are also not exactly, but they are found in microbes. So macrophages, what they do, they they identify those mannose particles in the microbes, and eventually they identify that microbes. So whenever microbes enters inside the body, they are recognized through the mannose binding proteins by the macrophages. So macrophages activated by M1 or classically active pathway, in which they stimulated interleukins like L1, L12, L23, and chemokines, and that causes inflammation. Or the macrophages, they start cytosis and killing by way of reactive oxygen species, lysosomal enzymes, and nitrogen uh, active species. So they causes injury and killing of many bacteria and fungus. Second pathway is alternatively activated macrophages, And this pathway is associated with the tissue repair and like we have got two pathways of macrophages. One pathway is killing the bacteria, is causing inflammation, but the other pathway is moving towards that repairing system. It has got anti-inflammatory effects. So this is again very important. The different pathways of macrophages and anti-inflammatory effects are occurred through interleukins 10, tumor growth factor beta, and growth factors beta, that is tissue repair. So it has got two simultaneously, it is performing two functions, that is the anti-inflammatory effect as well as the inflammatory effect. Lymphocytes again are very important. They produces CD4 T cells. They produces interferon gamma that eventually helpful in macrophages classical pathway, as we have seen this, right? Then they secrete interleukins 4, 5, and 13 that moves the macrophages for alternative pathway. And they secretes interleukin 7 and others to stimulate neutrophil. So this is the way how lymphocyte participates in causing inflammation. So um this is the way how it produces macrophages. They present antigens, right? Then T cells get activated, they produce cytokines, and activated T cells against causes activation of macrophages. So you can say this is a viscous cycle, like one thing is related to the other, one thing is stimulating the other. So in the way they are performing their function in complete harmony, you can see that none of the one cell is working alone. They have been teamed up to perform that are associated with granulomatous inflammation, like TB. TB is the one of the most common. Leprosy, we will also study leprosy, syphilis, cat scratch disease, sarcoidosis, and Crohn's disease. So at least you should know the names of these diseases and the causative agents that are responsible for granulomatous inflammation. Thank Any question you can ask?